Hello and greetings to all of you, dear friends. I hope you're all doing very well. I'm Milad Adi from bioinformatics.com, and in this series of videos, this being our very first one, I intend to explain the general steps and process of NGS data analysis, that is, next generation sequencing. I will present and explain these steps to you for free in the form of, let's say, 9 or 10 videos. I hope it will be useful for you. Given that I meticulously explain these complex videos to you in very simple, easy to understand language, I sincerely hope that they will truly and genuinely help you to effectively get started with your crucial NGS data analysis journey. All right, in this video, we want to go ahead and, well, make sure to watch the video. Watch this. Let's learn the basics of alignment and NGS data analysis together. As they say at the beginning of our journey, we need to first understand how sequencing is done, how sequencing is performed. Let's go ahead and watch this in the video. First, together, we have a sample prep. Take a look, guys. Yeah, when we receive a sample from a patient, let's say from a patient or any organism, we need to extract the DNA from the cells. Now, depending on what our goal is, we need to go ahead and target that specific objective. Whether it's DNA or RNA, whatever it is, we need to extract it. And then, since these DNA strands are very long and lengthy, we need to cut these long sequences. All right, now look, guys, these sequences. First, let me give you an introduction here, then it will explain sample prep to you. So, as you can see here, there's a white fragment that represents the DNA sequence of my patient. And on both sides of it, we attach two small DNA fragments during the steps of sample preparation before sequencing. This happens before sequencing. These small fragments are primers, and there are also some other fragments that are used during the sequencing process. But for now, just remember that these two fragments are attached to both ends of each DNA strand. Basically, there are two primary primers attached, which are also commonly referred to as adapters. And as I previously mentioned, they are meticulously made up of several other crucial parts as well, such as unique indexes and a few other essential components that contribute to their overall functionality and structure. But in a more general sense, we commonly refer to all of these components as adapters. And what you can clearly see here is a prime example of an Illumina flow cell, which typically contains 8 distinct lanes for processing samples. These 8 lanes are all located on a single flow cell, and essentially they are 8 channels where the patient's DNA fragments are loaded onto these specific channels, and these individual fragments are basically complementary to the primers that we attach to the patient's DNA fragment are attached to this flow cell. Now, as you can see, a patient's DNA fragment, a single strand to which we've attached primers on both ends, comes and attaches to both sides, binds to its pair, and then a step of what is called PCR takes place. A polymerase chain reaction occurs, and as you saw, the patient's original fragment separates. It leaves, and the copy of that fragment remains attached to the flow cell. So that fragment is washed out and leaves, and the fragment that remains attached undergoes bridge PCR. Polymerase chain reaction, commonly known as PCR, is carefully performed, and within a specific region of that particular lane, at a very small and distinct point, several intricate molecular bridges are formed. PCR or polymerase chain reaction subsequently occurs, and a substantial colony of that specific DNA strand is meticulously created in that precise region of the flow cell, specifically within that designated lane at that exact point, all facilitated through these intricate bridges. If you pay close attention to these PCRs, one step has just been completed. In this, during the cluster generation phase, something happened. It retained one type of primer and washed away the other type. It kept one model of primers and washed away and removed the other model so that it can proceed to the forward sequencing step to generate our forward file. Now, as you can clearly see, the crucial sequencing step is actively happening. And in all of those numerous fragments where the distinctive purple primer dye was successfully released, the sequencing steps take place by performing a series of regular PCRs using nucleotides that are labeled with fluorescent tags, and this sequencing is carried out. And each time a base is added and attaches, based on the reactions that occur, a light is emitted. And that emitted light serves as an indicator of which base, which nucleotide is present at that position. These lights are then interpreted by the sequencing machines to give us meaningful data, A, T, C, and G. Now, for each of them, look at that signal, which is like one part of the symphony of the cell. Just imagine it. Look, for example, here you have a colony of our DNA, that single strand of DNA that landed here. Due to the successive PCRs that have been performed, you now have a large yellow fragment here. You can see that the intensity of the light here comes and tells us with high confidence which base, which musical note is present here. In the read that is going to be sequenced from here, this base is a C, and as it moves forward, film. With the successive exposures taken one after another, you can see which bases are present at that point, or in other words, in that read. I'll skip over these parts. These sections of the video that you're seeing aren't really important at all. For now, you can leave these for later when you get to the main stages of sequencing, and once you become proficient in the analysis, you'll gradually learn those steps. Don't worry. Okay, if you pay close attention, something just happened. Look, guys, watch. Right now, a crucial and intricate step of bridge PCR took place, and a new, perfectly complementary DNA strand was meticulously formed. And when these newly synthesized strands are subsequently released from their templates, now the individual DNA strands, the free strands we have are both purple and blue. In the previous step, we had removed the blue strands. Now we need to remove the purple ones. See, now the purple strand has been removed. Now here you see just one example, but you should extend this to the rest of the strands that are attached to the flow cell, and then sequencing happens again, similar to the previous step, with the primer that is paired with the blue strand, meaning it is positioned opposite the blue, and in this way, sequencing happens again. And again, reading is done from those flow cells based on the signals that we obtained, considering the cameras that are present in the sequencing device. These are very powerful and provide the device with very high quality signals. The analysis of these and determining which regions each read covers is done automatically. This is performed by the sequencer itself, so we don't need to do anything particularly complicated in this regard. We just need to process them. And at the final stage, we receive the data. The data that we receive is in the form of a FASTQ file. Everyone in the upcoming videos will also get familiar with the FASTQ format. One of our free videos is dedicated to explaining the FASTQ file. But in general, keep in mind that the steps are sample preparation, cluster generation, sequencing, and after sequencing, we have data analysis, which, as they say, is very... this video has truly rendered the steps of sequencing and data analysis significantly much more straightforward for us. Our sincere thanks go to Illumina for generously uploading this informative video publicly on YouTube, and we are truly grateful that we had the valuable opportunity to access this video and were able to meticulously prepare our own version of the video for you. Until the next video,